first slide. I shall be talking about peptide nanoconjugates, and these peptide nanoconjugates are actually working in the application of biology. So I have given a title which is called Chemistry at the Interface of Biology. I shall be talking about three things as I discussed in my just last slide. First is the peptide-based self-assembled biomaterials, in which I shall be discussing about how exactly we have been able to synthesize new biomaterials for applications in different fields. Then I shall be talking about nanoconjugation-based drug delivery and cell internalization methods. Again, we have used different peptides for cell internalization applications, and then different sensing applications in the using different bioprobes, which includes peptides, aptamers, and others. Of course, because this is a time of lockdown due to coronavirus, and I was going through a few literature studies, and I found that basically a virus is made up of three things. This is peptide proteins, as well as nucleic acid and lipids. So I shall be talking a little bit for the students, specifically telling them about how these viruses are made, how this specifically the SARS coronavirus 2. I shall be just giving 3-4 introductory slides, and the answer for the coronavirus vaccines can also come from this. So it's a mix of our work and some of the things that might interest you in the recent context. So before I start, I just want to give you a brief background. I just want to introduce this topic to students, preferably by telling them about what exactly is a peptide. As you can see in the slide, peptide is actually a combination of different amino acids. The C terminal of one of the amino acid, which is a carboxylic acid, can combine with the N terminal of another amino acid to form this type of a amide structure by the loss of water molecule. This peptide is you can use any type of amino acids, whether natural as well as unnatural amino acids, to make different type of peptides. And once the size of peptide increases, it makes a protein. And once the protein is formed by combining different amino acids, the protein can take different conformations, starting from alpha helix, beta pleated sheets, and a lot of other conformations are also possible. But these are the major ones: the alpha helix and the beta pleated structures. In alpha helix, it's like a zigzag, this zigzag shape of molecules, monomers entangling together and making this type of form conformation. Whereas in a pleated structure, it's like a platoon, as the military platoons go after one another. So the peptides and the proteins also tend to cluster together and make these type of pleated structures. So once this primary structure and the secondary structure is formed, then these peptides can also constitute tertiary structure, where different alpha helix as well as beta pleated and other conformations can lead to the formation of a bigger protein structure. And these protein structures can also combine to form a quaternary structure. So this is a little bit about how exactly the proteins and the peptides can take conformations. Of course, there are forces that stabilize these protein structures, which include covalent bonds, hydrogen bonds, metal ligands, ionic interactions, disulfide bonds, non-bonding interactions. So these all make what is called as native conformation of a protein. Now, once this native conformation of a protein is formed in the body, it is very essential that that same type of native conformation is achieved even through the synthetic methods. We can now synthesize different proteins and peptides in our labs also. And but the important thing is that native conformation has to be achieved. Only then the biological activity will come. So with this background, I first would like to initiate our work that we are doing using these peptides and other nanostructures in biomaterials. So our idea initially was to start the self-assembly of these peptide structures or the protein structures from single molecule to supramolecular architects. The idea was to make the spontaneous organization of molecules to form a nano-sized or micro-sized supramolecular architectures driven by non-covalent interactions, which include different hydrogen bondings and other non-covalent interactions. So the different forms can be taken. Once a small protein or a peptide actually combines, it can actually lead to a formation of a fiber. It can lead to a formation of a tube. It can lead to a particle. It can lead to a matrix, and it can lead to a membrane. The most important thing is that the properties of these supramolecular structures is totally different from the monomers. And because of this, it actually leads to the formation. You can do a lot of applications which are totally different from a monomer applications. And a lot of work is going on to this. So we initially started working on this using different amino acids. We took natural amino acids as well as some unnatural amino acids, and then we of course worked on small peptides, and then moving to other chemically modified amino acids and peptides. So the plan of action was to take a monomer, combine it to form a supramolecular structure, which can actually lead to a different type of amino acid or peptide self-assembled structures, which can be a fiber, which can be a tube, which can be a vesicle, or which can also be a spherical structure. And then the most important point from the perspective of making this self-assembled structure is how exactly these are soluble in different solvent systems. Then chemical functionality is also very important, whether these are hydrophobic, hydrophilic structures, what type of interactions are there. So it ultimately leads to a formation of an architect, which could be then taken up for different applications. So we initially started working on this about 3 or 4 years ago, and we were interested in self-assembly of some of the aromatic amino acids. There was a report in literature in Science journal which actually talked about that different proteins in the body actually break down and into different amino acids, and these amino acids tend to self-assemble in the body and then tend to make amyloidic structures. Now these amyloidic structures are structures which are otherwise difficult to break, and they are toxic in the body. So they then lead to different types of pathological diseases, which include Alzheimer's, Parkinson's, and all other different type of other pathological diseases. So we actually started working on this outside the body system in the lab. We took up different aromatic amino acids, and we tried to self-assemble them. For example, we took phenylalanine, histidine, tryptophan, tyrosine, and we tried to self-assemble them using the biologically relevant and biologically viable systems. And we were able to make these fibril structures, histidine, tryptophan, tyrosine structures, which were actually characterized by NMR. Actually, Venus was one of the co-authors in this paper, if I remember correctly, Venus. And this was also then characterized using XRD, TGA, FTIR, and other studies. And we were able to understand how exactly these structures were making these different architectures. Interestingly, specifically, phenylalanine and tryptophan were showing very good amyloidic formation, and that led to our hypothesis that these type of amino acids definitely tend to make amyloidic structures, which might be having toxic effect in the body. So we were able to publish this initially, and then we are in a process of now doing in vivo studies to see how exactly they function in the body. So this was initial studies, and we also worked on some of the smaller peptide systems. For example, this dipeptide system with FMOC functionality attached at the end terminal, this particular system. We made a self-assembled structure, and we found that very nice spherical nanospheres were getting formed. And then we took up these nanospheres, and we incorporated different drug molecules, for example, ampicillin and others. And we then saw that these drug molecules were actually getting incorporated into this peptide nanosphere structures, and they could go into the body quite effectively. So this is the SEM analysis of these nanospheres, and we also did some biocompatible studies, and we saw that specifically ampicillin and doxorubicin were very nicely going into these nanospheres, and we could also release them according to our different environment available in the body, and we could show that these are actually getting cell internalized into the systems. So what I'm trying to tell you here is that even a small structure, peptidic structure, can be modified and can also be taken into the for different applications. The important point is that these are biocompatible systems. These are from the body. These are small parts of proteins which are taken up nicely by the body, and they are non-toxic and can also be used for different applications. So encouraged by this, we were also working on small systems in which we were trying to see what is the basic building block responsible for formation of different nanofibrils using amino acid structures. In this case, we took phenylalanine, and we found that a dimer of phenylalanine is actually responsible for making these small, this red shaped nanodots, which actually combine and form this type of fibril structures in this phenylalanine structure, in this phenylalanine self-assembled structure. So a phenylalanine dimer makes a nanocylinder, and that nanocylinder is responsible for a nanofibril. We did extensive characterization studies. We found that phenylalanine molecules organize into dimers of size of about 1.78 nanometers, which were actually characterized using DLS and SAX studies. And we also found that these dimers include the hydrogen, the stabilizing factors in these dimers is because of hydrogen bonding and cation pi interactions. And these phenylalanine dimers go through secondary self-assembly processes to form beta sheet type of nanocylindrical structures. And the photolimisis of these amyloidic nanofibrils originate from the nano dimensional clustering of zwitterionic bridges. This is in fact, interestingly, this is the first study which actually talks about the interactions between the small amino acid structures leading to the self-assembled architecture. And we were able to recently publish this in Soft Matter. So we are working on this further, and we are trying to see how exactly peptides and bigger proteins can also generally tend to make these type of clusters by studying their characterization effects. So apart from these slightly more structural implications, we also are working on biocompatible short peptide based approach for water treatment. Specifically, we are working on removal of cationic dyes. And in this case, this is an example in which we have synthesized this particular dipeptide, which has led to the formation of these type of self-assembled structures, which includes the fibrils, needle type of structures, and something in between, and depending upon what type of experimental conditions have been used. And we have then treated these peptide fibrils with different dyes, and we found that specifically the cationic dyes, which includes crystal violet and methylene blue, were able to get treated. Actually, these peptide fibrils absorb these dyes on its surface and lead to the water treatment, which actually converts a dyes contaminated water into a clean water system. Interestingly, this particular peptide fibril was able to absorb 625 milligrams per gram of crystal violet, which has been the best reported so far in literature. Encouraged by this, we actually we synthesized different peptide systems. Now, I just want to introduce peptides for the sake of students here. The difference between a peptide and a peptoid is that the peptoid has the side chain of a peptide on its nitrogen rather than on the carbon atom. So the structure is almost the same. The difference is that the side chain is on the end of nitrogen, which actually is more stable because this nitrogen is able to he is able to hold this particular molecule in a much more tighter form, leading to the formation of a structure which is slightly different from peptide, but structurally more stable as compared to peptide. So we were able to synthesize different aromatic peptoids and using this chemical strategy. And these different peptoids were then also tested for the treatment of different dyes in water. And we found that these cell peptoids were showing very nice self-assembled structures in form of fibrils, needles, and different tubulins. And we found that because of the structural rigidity of these peptoids, these fibers were even more stable as compared to the peptide fibers. And these were also showing very selective adsorption for cationic dyes, and in specific for crystal violet and methylene blue. And this work has been submitted in Chemical Communication, and we're waiting for its approval. Okay, so this was one part. I just want to have given you an idea about how exactly we are working on peptide-based structures which are taken up in biomaterials, and we are working on some other aspects, but due to paucity of time and because I have some other slides to show you, I'm now shifting towards the cell internalization applications. So for cell internalization, we were interested in nanoconjugates, and for nanoconjugates, it's very important to understand what is the property of a nanoparticle and how exactly it behaves when it is interacted with different molecules, specifically biomolecules like peptides. So there are different aspects of a nano system. One of the thing is the size, which actually ranges from 1 nanometer to 100 nanometer, and you can actually modulate that size depending upon how exactly you are synthesizing the nano system. And then there are the surface modulations that you can do on nanoparticle surface, depending upon whether it's positively charged, negatively charged, or what is the shape of the structure of the nanosystem. So you can do the pegylation. You can actually do surface functional, attached surface functional groups such as thiols, amines, carboxylic acids, and then you can also modify the surface charge. And depending upon what type of target ligand you are approaching, whether it is a peptide, antibody, or aptamer, which is a DNA strain, you can actually modify the nanosystem quite effectively. Then it's very important to understand what is the shape of the nanosystem. Now the shape is important because you can actually increase the adsorption on the nanosystem depending upon the shape. So you can have a sphere, you can have a cube, you can have a rod, plate, star, or other type of shape systems depending upon what is the experimental condition that you're using. Then what type of material is being uploaded, whether it is a protein drug conjugate, dendrimer, peptide, or hydrogel system, or what type of system you are working upon, you can actually modify the nanoconjugates. So these are the major concerns when you are actually approaching to make a nanoconjugate system. So coming from a chemistry background, is very important to understand what are the interactions that you are looking at. So depending upon the nanoparticle, you can have an interaction which can be a chemical in nature, which can be a covalent, for example, gold nanoparticle sulfur chemical bond system, or you can have a specific enzymatic interaction also between a nanosystem and a biomolecule. Then you can have a diffusion based functionalization depending upon what is your biomolecule system. And then of course, electrostatic surface modification, which is being held together by positive negative charge. So we were interested in peptides and amino acids to get it conjugated to nanoparticle systems. Specifically, we started with the metal nanoparticle systems. Initially, we started with gold nanoparticles, and we studied different amino acids that can be conjugated to these gold nanoparticle systems. The red color here shows the one which were actually able to reduce the gold nanoparticle surface and were able to get conjugated to these gold nanoparticles. Whereas the blue ones are the ones which were not showing any combination. So we actually concluded in this paper that it is because of the specific interactions of the side chains as well as the N-terminal and C-terminal interactions with the gold nanoparticle, which are leading to the formation of nanoconjugates between gold nanoparticles and amino acids. And similarly, we also studied with a few later, we studied with this silver nanoparticle system and found that some of the amino acids were actually conjugating quite effectively, whereas others were not doing as effective as some others were. So one of the major reasons for this is the structure, as I have discussed, the side chain of the amino acids. The other one is what is the pH of the solution that the amino acid is leading to, and there are some other considerations also, and these works were published in different journals. So we moved from amino acid, we actually studied some peptidic structures, small peptides we took up with these metallic nanoparticle systems. We first made these metallic nanoparticle systems using citrate, and then we loaded these peptides on these metallic nanoparticle systems, and we were able to synthesize monodispersed and spherical gold nanoparticle systems using these nanoparticle peptide conjugates. And we also studied their antimicrobial activity, and we found that the antimicrobial activity was better than both peptide as well as the nanosystems, specifically in the case of silver nanoparticles. So we moved forward, and we were actually interested in studying what is the quantification of the molecules on a nanoparticle system. So we specifically took citrate ions, and this paper has been recently published, and we found that specifically the exact number of molecules on a nanoparticle surface can be quantified using TGA, which has been reported for the first time. So what we did, we took the nanoconjugate system, which was between gold nanoparticle and citrate. We subjected it to TGA analysis, and we found that there are different loss of weights depending upon the temperature as it was increased. So first there was monocitrate ions going, then there was tricitrate ion going, depending upon at what pH you are doing the studies, and we were able to calculate the total number of citrate ions on the gold nanoparticles specifically. I would like to specifically highlight here that we did this work with Professor DVS Jain, who is not us with us more anymore, and it was his idea actually. He started discussing this with us, and we were able to finally get it published this month only. And this is a work that is in tribute to his memory. So, I mean, we were working on these nanoconjugates where we were specifically discussing about metallic nanoparticle systems. So one of the proposals that we put up to DST was that we can actually synthesize these nanoconjugates where we can conjugate a cell penetrating peptide. Now these cell penetrating peptides are specific to to some cells, bacterial cells or some microbial cells, even some viral cells also. There are antiviral peptides also, which can actually be conjugated to these gold nanoparticles to make this type of conjugate system, gold nanoparticle cell penetrating system. And then we can load a fluorophore, for example, an FITC, to make this type of a conjugate. And then we can introduce this into bacterial cell and specifically internalize into the bacterial cell in the presence of the host, which might be a mammalian cell, because the cell composition of mammalian cell as well as compared to the bacterial cell is different, and these cell penetrating peptides tend to go only into the bacterial cells as compared to the mammalian cells. So we started working on this and we were able to synthesize these gold nanoparticle conjugates with cell penetrating peptides using both electrostatic as well as covalent approaches, and we found that these were actually getting internalized, and we have published this. But we were not very happy with the results, because as you can see from this SEM analysis of these microbial cells, that only a very few of these conjugates could internalize. The most of the microbial cells or the bacterial cells were not, these were not able to internalize into those microbial cells. So one of the major reasons for this was the introduction of FITC here. The FITC actually leads to the conformational modification of the peptide, which tends it to make it inactive, and it doesn't allow it to go into the cell as effectively. So we thought of a solution to it, and we switched over to quantum dots. Now quantum dots are semiconductor nanoparticles which are in itself fluorescent. For example, gold nanoparticles are not fluorescent. They need additional tagging on it, whereas quantum dots are itself highly fluorescent and they don't need an extra tagging. So we worked upon this cadmium telluride based quantum dots, and we changed the scheme in which we took specific cell penetrating peptide, for example, this cell penetrating peptide, conjugated it with the quantum dot which was synthesized in our lab, and then made this type of a conjugate system. Then in one of the cases, we just introduced into the bacterial cell, and in another case, we actually loaded the drug molecule on it, because we wanted to increase the efficacy of drug molecules, because if the drug molecule will be targeted, it will go directly into the microbial cell. It can have much better efficacy at a very low dose, and we studied this type of phenomena. So for that, we first conjugated these quantum dots with cell penetrating peptides. The quantum dots were having a zeta potential of -36.1, whereas when this conjugate was made, because of the positive nature of the peptide, the zeta potential was decreased to -28. And we also understood the conjugation using UV visible analysis, and we found that there is a quenching when the quenching of the fluorescence of the quantum dot when the peptide concentration was increased, as well as we did the gel electrophoresis to see that as the positive charge of the peptide is increased, the gel of this conjugate moves much faster towards the positive electrode. So with these analysis, we did the internalization studies again, and we found that there was much effective internalization in this case, and we were able to internalize into different bacteria, both gram positive and gram negative bacteria. And this work has been now published recently And we are working upon using different cargoes for these type of nanoconjugates, for example, DNA strains and others, to see how exactly we can use it in different applications, apart from bacterial internalization. So this is one of the work that, another work that I have discussed. So now as initially I discussed, that I shall be talking a little bit about the present problem, that is coronavirus. So before that, for the sake of students, I just want to introduce the nucleic acids to them, the nucleotide structure. Most of you might be knowing that a nucleotide, whether DNA or RNA, has three type of constituents. One is the phosphate group, the other is the sugar, which can be a ribose or a deoxyribose, depending upon whether it is an RNA or a DNA. Then it can be a base. Base can be of two types, purines or pyrimidines. And of course, everyone knows purines are adenine and guanine, and pyridines are cytosine, thymine, and serosine, depending upon which nucleic acid we're talking about. Specifically, in the case of RNA, the difference between RNA and DNA is one of the difference is the sugar. The C2 of the ribose sugar is having hydroxyl group in RNA, whereas in DNA it is only hydrogen present here. This is one of the difference. Another difference is the base. In RNA there is uracil instead of thymine. So these are the two differences. Of course, everyone knows about the double helical structure of DNA, which actually was developed by Watson and Crick, and this has a intermediate multiplicities. There is a minor group, there's a major group, and then there is a middle. So this is about DNA or RNA. I just wanted to give a couple of slides about lipids also, because I am introducing the virus here, so it is very important to understand all of its constituents which are there in this. So lipids are actually compounds which are not soluble in water, in simple terms, and they are generally soluble in hydrophobic solvents. And there are different type of lipids. One is the glycerol based. They can be fats, they can be phospholipids. Specifically, I want you to focus on phospholipids, because these are the ones which are actually making the cell membranes, whether they are the cell membranes of microbes, or they are the cell membranes of our body, mammalian body, or others, depending upon their constituents. For example, in human bodies, sphingolipids are more than phospholipids. Sphingolipids are positively charged. So these phospholipids, in these phospholipids, one of the fatty acids of glycerol is replaced by a phosphate group. And phosphate group is slightly more, it's a negatively charged, so it has a charge. So that's why it tends to like water. So it is less hydrophobic as compared to the normal fats. So these are the phospholipids which make this type of a bilayer system. And this bilayer is then leads to the formation of cells. So coming to the structure of SARS coronavirus, as you can see in this slide, SARS coronavirus comprises of this lipid structure. This is green one is the lipids bilayer structure. Then there are glycoproteins. Glycoproteins are proteins with some carbohydrate molecules on them. Then there are some membrane proteins in here. And then of course there are some proteases structures again, which are coming out of these viruses, which actually are the targets which can actually take out something from outside and take it in, or the vice versa. So all these viral strains, including the SARS coronavirus 2, are made up of mainly these three components: the proteins, which includes the glycoproteins, then the nucleic acids, and then there's the lipid bilayer. So why I'm discussing this is because, as you can see in this slide, last month only in Nature, there was an article in which talked about coronavirus vaccines. It stated that there are many lead molecules as far as vaccines are concerned, and different groups are working on it. But specifically, there are 4 types of vaccines which are going to come up. One is the viral vector vaccine, in which a virus such as measles or adenovirus can be genetically engineered to produce coronavirus proteins in the body. These coronavirus proteins will then work on the coronavirus SARS-2, which actually leads to the killing of that virus. So that is one of the approach that has been taken up. The second approach is protein-based vaccines, because there are glycoproteins. They can be inhibited by a different target, which also includes a protein, or a peptide-based vaccine. In this, many researchers are injecting coronavirus proteins directly into the body. The fragments of the proteins or the protein shells that mimic the coronavirus outer coat can also be used. So they are actually using a lock and key hypothesis for blocking the proteins on the virus by using different other proteins. And there are small protein subunits which include peptides, which are also being used. Then there is a nucleic acid vaccine which is coming up. So the researchers are aiming to use genetic instructions like DNA or RNA for coronavirus protein that can prompt an immune response. The nucleic acid is inserted into the human cells, which can then churn out the copies of the virus protein. Most of these vaccines encode virus spike proteins. So again, nucleic acid vaccines are working on these proteins or the glycoproteins present in the virus. Now, the most interesting thing is that out of all these candidates which are already in the market, the most worked upon one is the protein-based one, which includes more than 30 candidates which are there already. Some of them going into clinical trials, some of them will be coming up soon. And then followed by the viral vector vaccine, and then followed by the nucleic acid vaccine. And of course, there are other chemical vaccines also that might be used. So what I'm trying to tell you here is that the solution to this problem actually lies in the structure. And the structure is composed of these 3 or 4 important components. So if we understand the virus properly, we can actually come up with the solution. So just wanted to, because I just didn't want you to only focus on our work, just wanted to tell you that these type of work can also be used to work upon these vaccines. We have submitted a project, and we hope that it will be funded. And so we would be working on this if it is workable, if it is funded. Okay. So finally, another application that we are working upon is biosensors. Now, we were initially interested in the sensing of pesticides. As you know, in India, the pesticide toxicity is a big problem. There is a non-rational use of pesticide, which leads to a lot of problems. Specifically, we are focusing upon organophosphate pesticides. Now, these organophosphate pesticides inhibit enzyme acetylcholine esterase, which is a neurotransmitter, thereby causing symptoms like weakness or paralysis, and they are highly toxic to vertebrates and also to other species. So this organophosphate phosphate pesticides include parathine, malathion, phorate, and other pesticides. Now, the major problem with the detection of these pesticides is that generally we use conventional techniques such as HPLC, gas chromatography, mass spectrometry, and flow and antibody-based amino acids. Now, the major problem with these techniques is they are lab-based techniques. They are sophisticated instrumentations. They need skilled manpower, and a lot of time is consumed in this, and a lot of complex sample pre-treatment is required, which makes it quite expensive and time-consuming. So we were actually working upon these nanoconjugate systems, and we thought that these nanoconjugate system has a very specifically in the case of gold nanoparticles, they have a very nice SPR, and they can actually change color quite easily when they're aggregated. So we initially started working on a detection of these pesticides, and we took up acetylthiocholine. Acetylthiocholine then actually interacts with acetylcholine esterase enzyme. It forms thiocholine. So this acetyl group is cleaved and the thiocholine is formed. But when this organophosphate pesticide is there, it interacts with this enzyme and blocks this enzyme. So now this acetylthiocholine cannot get converted into thiocholine. So these are the 2 pathways, one when the pesticide is present, another when the pesticide is not present. So when the pesticide is not present, thiocholine is formed. Now we used a cysteine capped gold nanoparticle system. Now this cysteine capped gold nanoparticle system is negatively charged, and this thiocholine is positively charged because of quaternary nitrogen. Now these make a very nice electrostatic interaction and lead to the formation of this type of aggregate, where this gold nanoparticle, which was earlier red in color, gets aggregated because of thiocholine, and the color changes to blue. Now, if the pesticide is present, thiocholine will not form. The gold nanoparticle will remain as such, as red in color. Whereas when the acetylcholine esterase is not blocked by the pesticide, or when the pesticide is not present, the color would be blue. So it's a very, as you can see in these vials, the color changes quite effectively. So you can actually detect the presence of an analyte, which is pesticide here, by only changing the color. by adding these solutions. So this was the first work that we published, and it was taken quite well. So we moved on. We took some other pesticides such as phorate in this case, and specifically used aptamer conjugated gold nanoparticles. Aptamer is a single-strand DNA molecule which has a specificity towards a particular analyte. For example, there are pesticides that we can make aptamers for specific pesticides also. So we took gold nanoparticles, we synthesized gold nanoparticles, we conjugated them with aptamer phorate, with aptamer, which was specific for that pesticide, because the aptamer was having, and there were positively charged as well as negatively charged subunits on it. It got conjugated with this gold nanoparticle. Now, when the phorate was added to it, the phorate took away the aptamer, because aptamer was specific towards the phorate molecule, and the gold nanoparticle was free. Now, when we add an electrolyte like NaCl into it, it led to the aggregation, and the color changed to blue from red. Whereas when there was no phorate present, the complex of aptamer and gold nanoparticle remained as such. And when the electrolyte was added in this case, there was no aggregation and the color remained red. So in the case of pesticide, the color became blue, whereas in the case of, in the absence of pesticide, the color remained red. And the LOD, which is called as limit of detection of that particular pesticide, was quite effective. That was 0.1 nanomolar. That means you can actually detect 0.1 nanomolar of the pesticide using this technique by just changing the color by adding these constants. So this was also taken quite well. And further moving on, we took up malathion, which is also another organophosphate pesticide. In this case, we used polymer, aptamer, and gold nanoparticles. In this case, we took the aptamer. When the malathion was present, it got interacted with this pesticide to form this type of conjugate. And then we took a positively charged polymer. Now this polymer is not because it is a positively charged, and the conjugate is already formed between aptamer and the malathion. So this polymer remained free. Now when we added the gold nanoparticle, which was negatively charged, it got aggregated by the polymer, and the color of the gold nanoparticle changed to blue. However, when the target was not present, the malathion was not present, the aptamer and the polymer conjugated to make this type of complex, gold nanoparticles free, and the color would remain red. The LOD in this case was found out to be up to picomolar range, which is considered as quite good, and it was published in Biosensor and Bioelectronics, which has an impact factor of more than 9. Further moving on, we instead of using a polymer, we used a peptide. Now this is the first report which has come out for these type of sensors using cationic peptides. So instead of a cationic polymer, we use the peptide, which also conjugated with the aptamer quite effectively. And when the gold and this pesticide was not present, the color was, this is the actual image of the solution. When it was not, the gold nanoparticle was as such when there was no pesticide, and when the pesticide was added, the color changed to the blue. And this was quite well taken up in Chemical Engineering Journal. It came on the cover page of the Chemical Engineering Journal, which has an impact factor of more than 8. Okay, so apart from these colorimetric techniques, we also worked upon fluorimetric techniques, in which we use quantum dot based sensing technique. And the quantum dots, because as I have already introduced, they are fluorescent molecules itself. So quantum dots, the fluorescence of the quantum dots can be quenched if we use a positively charged entity. In this case, we took a positive polymer, which led to the quenching of these quantum dots. And now, depending upon whether the pesticide was present, the quenching happened because the polymer was free, because pesticide actually interacted with the aptamer, and the polymer actually leads to quenching. Whereas when there was no pesticide present, the polymer as well as the aptamer were free to interact, made this type of conjugate, and there was nothing to get conjugated to quantum dot, and the fluorescence remains as such. Now fluorescence is quite a sensitive technique, and the LOD achieved in this case was 0.06 picomolar. Apart from pesticides, recently we published a paper in which we sense the metal ion, specifically the lead ion, using plasmonic gold nanoparticles and cationic peptide based sensor. In this case, again, we used the aptamer, which was specific for the lead molecule, lead ions. When the lead ions were present, the aptamer got conjugated with the lead ions, and the peptide, when was added, remained free, and that peptide actually conjugated with the gold nanoparticle, leading to the aggregation, and the color changed to blue. Where when the lead ions were not present, the aptamer was free as such. It conjugated with cationic peptide, lead to this conjugate, and the gold nanoparticles were free, and there was nothing to get interacted with them, and the color remained red. And we actually did some interference studies in the presence of different metal ions and found that it's quite specific towards lead ions, with LOD up to 98.7 picomolar, which is the best reported so far for the detection of lead ions. So we are also working on some of the bimetallic nanocluster systems now, in which we are trying to understand their sensing platform for different analytes. Specifically, we have worked on ethyl parathane. This work is still under consideration and showing very good results with limit of detection up to picomolar level for this. In fact, we by using these type of systems, we can actually do even better. We can do much more sensitive detection of other analytes using this type of system. So this is another work that I just wanted to show you. So I mean, encouraged by this, we actually submitted a project to make a kit for these type of sensors, and we were lucky to get funding from DST under Technology Development Program, in which they our partner is from IIT Madras. They have been able to give us funding to make this type of kit, and we are working on this also. Finally, this is my conclusion slide. So as I discussed, we are working on basically three things: peptide-based self-assembled biomaterials, in which we have been able to first understand the different biomaterials, how exactly they are relevant in pathological diseases like Alzheimer's, Parkinson's, and also we are working upon some self-assembled systems for water treatment. Then nanoconjugation based drug delivery and cell internalization, where we have used these biocompatible nanoconjugates for pH responsive drug delivery vehicles for different cargoes. Still, this is a work in progress, and we are hoping that we'll be able to get better results in future. Then, of course, as I discussed in the end, sensing using bioprobes, where we have used peptide and aptamer-based biosensing for different analytes, and with quite high, quite ultra-sensitive detection limits. Finally, just give you an idea that this biomolecule-based coronavirus vaccine, which is the most likely candidate for coming coronavirus vaccine, includes these type of systems. And of course, we have to do a lot of things, computational designing and others, to understand the real thing. So this might be one of the candidates which might be able to solve this problem. Finally, I like to acknowledge, of course, this work is not possible because of the students. Only it is the hard work and the dedication of the students which has led to these results. And specifically talking about pesticide sensing, Rajni has been the instrumental force. Then Munish worked upon the cell-penetrating peptide nanoconjugates. Surinder is working on peptide self-assembly. Prabjot is working on amino acid self-assembly. Priya is working on peptide drug delivery. Deepika is working on biomolecular nanoclusters. Gurpit is working on peptide nucleotide sensing. And then, of course, there are few who have already left. I have not discussed about their works because of the paucity of time. Last but not the least, I'd like to thank our collaborators. The most important one is Dr. Nishima, who is actually the backbone of the nano work, which we are working in our lab. Then Professor Rahul Jain, who has always supported us. Then, of course, some international collaborators with whom we are working upon different applications. And lastly, I'd like to thank the funding agencies for giving us the funds. Thank you very much, and I look forward to the questions. I hope I'm on time, Venus.
Yeah, you are perfectly on time. Very nice lecture, and it was fantastic to see all the things that you have been able to do with all the constraints which we have in our places. This is really, really fantastic. And
thank you.
Yeah, so I request the audience to raise their hands if they have a specific question for Dr. Rohit. And you can raise your hand there is an option in the Zoom app to raise your hand. Or you can also write your question in the chat box and I will ask it on your behalf. So please raise your hands if you have any specific questions related to the work which Dr. Rohit presented here. In the meantime, I have one question for Rohit. In the cell internalization work which you discussed, so I mean, you chose nanoparticles and which are coated with the peptide, which assists the internalization of the material.
Mhm.
So what are those peptides basically? I mean, how are they being, I mean, sensed by the cell? I mean, what kind of internalization is there? What is the mechanism of that thing?
Um, the idea of these cell penetrating peptides actually started coming up from TAT peptide, as you might be
Okay.
aware, that so these are actually small cell, these peptide fragments of these TAT peptides,
Uh-huh.
which have been able to interact with the glycoproteins as well as
Okay.
some of the receptors present on the cell membranes of these
Okay.
microbials. And because there is a difference in the cell, this difference in the constituents in the microbial cells,
Mm-hmm.
which is based more mainly on the phospholipids, whereas
Okay.
in the case of mammalian cell, it is more mainly on the single lipids. Single lipids are more positively charged, whereas phospholipids are more negatively charged.
Okay.
So these peptides take a conformation. They are generally slightly bigger. For example, the one that we took for the studies for cell penetrating peptides
Uh-huh.
were using quantum dots was about 14 amino acids, and one was about 11 amino acids. So they tend
Okay.
to take conformations. For example, I'll show you.
So they were alpha helical as you have shown in the slide. They looked alpha
Yes,
helical.
they were pseudo alpha helical. They were not
Yeah.
because their size were not that big,
They are too
but
short
they
for a proper alpha helix, I guess.
yes, yes. But they have a conformation which actually leads to an interaction between them and the bilayer
So basically, the
glycoprotein.
internalization is based on a particular sensing site, not it's not a in. I mean, it's not a very general internalization. So some
Yes,
particular glycoprotein or some other sensor is sensing that peptide sequence and then
yes.
assisting the
There
internalization.
is still a big question mark about the mechanism of these cell penetrating peptides, how exactly they do. Some of
Okay.
them, some of the reports tell that they are actually able to internalize into these cells using diffusion method. Some of them
Hmm.
actually work upon this other hypothesis using carpet method and others.
Okay.
But the good thing about these cell penetrating peptides is that it is different from antimicrobial peptide, because
Hmm.
antimicrobial peptide tend to kill the cell by acting
Yeah,
upon the,
disrupting the membrane.
whereas
Yeah.
these actually diffuse through these like
Hmm.
an external molecule going into the cell.
So what is the
They are
size of the whole assembly, the
that's
nanoparticle plus the peptide coating?
so that size is very important. For example, the quantum dots that we synthesized had to be less than 3 nanometers, preferably 2
Okay.
nanometers, or even less than 2 nanometers if possible, 1.5 or 2, because
Okay.
the pore of the bacterial cell is about 3 to 5 nanometers. So you cannot make a conjugate which is more than 5 nanometers or more than 10 nanometers. So for that, actually, we took a lot of time. So because we were able to, we had to use different. I have not discussed the specificities of how
Okay.
exactly we have synthesized. That can be found in the papers.
Yeah.
But we were able to make 2 nanometers size, 2 to maximum
And with
2.5.
the coating, the final size would be around.
The
Uh,
final size was because these cell penetrating peptides are actually smaller than the nanoparticles. They are in the
okay.
angstrom region. So that was about 3 to 3.5, not more than 5. So it was less than
And
5.
what happens to them after? I mean, ultimately they will be endocytosized, and then lysosomes will try to uh, denature them. So what
Yeah.
happens after they are internalized? I mean, how?
So,
I mean,
so far we
what
have
is the
been,
lifetime of these things inside the cell? How long they stay intact?
we haven't checked it because I think we need a biological support for this. We need a
Okay.
person who can have actually do these studies. Right now, we are able to internalize them. We are trying to use different cargo systems. For example, we have used drug molecules. We have used some proteins already, and we
Okay.
are working upon some nucleic acids. If we can insert some DNA strands or something like that. But of course, it's
Okay.
a very relevant question that you have raised. We need some sort of, we need to understand them by using some collaboration, I think. So we are working on this. There is a group which is taking quite an interest into this work, but it's still preliminary stages, so I cannot comment on that.
Okay. Okay. Yeah. So I request the audience once again, if they have some particular question for Dr. Rohit, if they want to ask, they can raise their hand. And yeah, so in the meantime, so there is another thing about the virus that you discussed. So there are uh, drugs also, which basically interfere in the mechanism. There are two types of drugs which are available at the moment, antiviral drugs. The ones which interfere with the, uh, I mean, cell fusion of the membrane of the vesicle of the virus and the membrane, which is basically the endocyte, which is inside the cell. So that there is one vaccine which targets the M2 protein and blocks that fusion. Not vaccine, it's a drug. And then there are second drugs which blocks the release of the virals from the surface of the cell so that it doesn't go into the next cell. So there are already two, how different when you are saying that you want to, basically work on developing peptide based methods or vaccines. So what about the drugs? Can't we have a drug also? I mean, shouldn't we be channelizing our effort in having a drug rather than a vaccine? Because the problem with the vaccine is any vaccine which we have at the moment, it gets outdated after a year. So why can't we just have like any other disease? Why can't we have just a drug which targets the virus specifically?
So the one that the drug concept that you raised is quite pertinent. And actually, apart from one drug, there might be multiple drugs also. They can be repurposing of different drugs. So it depends. I mean, what I'm talking about here is the proteases structures. There are some proteases which actually come out of virus, which have specific structures. So if we block those proteases, or somehow we um, make a molecule or vaccine which can actually lead to the blocking or maybe inhibiting the conformation or changing the conformation of those proteases, we will stop the multiplication of virus as such. So that is the principle on which it is based. But of course, as you have discussed, there are other
Yeah,
approaches.
because there is a lot of, I mean, these there are two types of drugs. I mean, as I said, so these are there in the market, but obviously they are not generic. The virus, it keeps on changing its machinery and they do not remain generic. So that's the problem. But
Yes.
I mean, right now the whole world is working on this thing, so they
So,
will definitely come up with the
so
solution to this problem.
I wanted to tell you here was that this actually came up in the last week of April itself in Nature.
Yeah,
It actually discusses what exactly is
the
happening right
approaches.
now.
Yeah, the
So
approaches
it
which
states that about 100 groups are working
yeah.
on these type of structures, on these type of biomolecular structures, and out of those, uh, these are mainly divided into three parts. One is the
Yeah.
protein base, the other one is the nucleic base, and then of course the viral vector base, and some others chemical based also. So what I was trying to tell you is that the solution lies in the problem itself. Let the
Yeah.
structure it, and of course we need a molecule which is should be biocompatible with the body.
Yeah.
So we have to see the problem with the other organic ligands is that we have to really see their toxic effects,
Yeah.
whether they are be able to they might be having very good antiviral effect, but they might also lead to other side effects. So
Yeah.
in that context, we were actually, I was just trying to propose that this type of structure analysis can be done. For example, we can understand already there are papers in which the X-ray crystallography of some of the
Yeah.
viral structure has been already done,
Yeah.
which actually tells you what is exactly the conformation of these proteases. So depending upon that, you can actually propose some structures using some modeling methods and others also. Of course, there is a big question mark whether what you propose from modeling is going to work in the lab or not. But these things can be done, and then using these type of biomolecules which will be biocompatible to the body, we can actually come up with new molecules. Of course, this is just
Yeah.
just wanted to
No, that's a good.
inspire
I was
this.
just giving my
Yeah,
inputs
yeah.
into that. Okay, so I don't think so. Students have any specific question. So I just like to, which is a very common question for our students, for most of the people, is that most of the students that we work in chemistry departments, and you also work in a chemistry department, and most of the work that you discussed, people are somehow not, have a notion that how will they learn the biology aspect. I mean, it is not related to your talk. I just want you to tell the students in that way. So, what is your take on that? How? Uh, I mean, how? how a chemistry student adapts with the kind of biology you are doing. I mean, yeah. So if somebody wants to do research in the fields in which you
I'll
are
tell you.
working, or if somebody wants to join your group in the future, so what kind of expectations will you have? I mean, and how easily will he adjust or not?
So it's a very, very important question, because when I joined our department here in Panjab University, first, one of the major problems was that the students were not coming. They thought that they have to do biology, and they will end up not doing chemistry at all. So what I want to clear is this, that this is all chemistry, whether it is self-assembled part, whether it is nanoconjugate system. We are just doing the applications. And just to inspire students, I just want to tell you them that out of the Nobel Prize which has come in the biological sciences in last 10 years, I think if I'm not wrong, 3 or 4 of them are from the chemistry background people who have been able to. So one thing is very clear, that people with good basic chemistry knowledge can easily work in biology, because there is a difference in the way these two streams are taken up. For example, I myself have done biological screenings and have understood the other biological mechanisms, because in biological studies there is a set protocol. You have to do this. If you have to do incubation overnight, you have to do incubation. You cannot do much about that. Whereas in chemistry, there are hundreds of things that you have to look at when you're putting up.
Basically, what you are trying to say is a chemistry person can definitely do biology, but a biology person might be difficult for him to do, or him or her to do a chemistry.
I don't want to be sounding controversial here, but I'm not saying the opposite. But yes, definitely I'm saying that if you understand the basic chemistry well, and if you have an idea about, even if you have not studied biology in +1, it doesn't matter at all. In fact, I have seen people coming from a mathematical background doing much better in this, because they have a clear mindset in this. So it is possible reversely also. So what I want to tell students is this, that don't worry what application you're looking at and what you have done already. It is more important that you understand the problem effectively, and you see that what things need to be done, and this is all possible. So even in our studies, we had collaborators who are immunologists, who are biologists, who have a very strong background in biology. They are working with us on this, and we are just getting in. Of course, I have understood many of these things by discussing with them regularly. So the future is for this interdisciplinary science. You cannot just sit in your room and just say that I will do this and I will not go beyond this, and I will not like to understand this because this is not my field. This is not going to happen in future like this. Even if you don't understand, you have to discuss with people. You have to see what is the view and how your work can be applied into that work. So that type of mindset should come into the students from the beginning. So they should understand. Of course, liking and disliking is also another thing. If you're interested in that topic or not, that is a separate thing. But you should be open to the ideas. You should understand, okay, this is the way we can do, this is how we can go ahead. So an example is that we are able to do so much applications only because we are open to it, and still we are open to it. We, in fact, if you remember, Venus, I think when we discussed this self-assemble part, you just told me that this is the thing which is possible with this NMR. So I was not knowing that before you. So that thing happened only when we discussed and we were open to the ideas. So that is the bottom line that I want to tell to students.
So basically, I would like to summarize it in this way, that what Rohit is doing in his lab is, I mean, it might sound like a biology. It is biology, but it is it is chemistry of biological molecules. So it is chemistry of just a bit complicated looking molecules. And I have a question. I have got a question for finally. Yes. So we have a person who wants to, I can't pronounce the name because it is written in some other language. So I'll request the moderator to put the person on screen so that he or she can ask the question.
Okay. Hi. Can you hear?
Hello. Bye. Hi. Yeah, we can hear you. Please introduce yourself and ask your question.
Yes. I'm from Japan.
Oh, welcome.
Yeah. Hirona Bahojo. How are you doing?
I'm doing fine, Professor. How are you, Professor?
Oh, it's good. I have a question about self-assembly of a dipeptide.
Yeah.
You talked about phenylaldehyde.
Yeah.
And in some cases, it forms a spherical particles, and the other cases it's a fibrillary structure. So how can you control the shape of the molecule?
Yeah.
Aggregates.
Yeah. So I just want to introduce Professor Hojo first to the audiences. He's actually one of our collaborators. We are having, and I'm very happy. I was not knowing that he's coming. We are having a Spark project with him actually, and he has been to India. I hope you are doing fine in Japan.
Yes.
Okay.
I'm fine.
So to answer your question, Professor Hojo, in this paper, we were actually able to self-assemble small these phenylalanine structures into fibril network. As you can see in this slide, this is the network that was formed when the phenylalanine was able to self-assemble to form these type of fibril network. What we have proposed here is that these fibril networks are actually combination of small dimers of phenylalanine. As you can see here, these small phenylalanine dimers actually are the subunit of this fibril network. They tend to cluster together. We have, apart from other studies, we have done SAC studies where we have actually found the angles which are or the distances between these phenylalanine monomers, which are making them to make a dimer, this type of a dimer. And this dimer actually gets, this dimer then joins together to form this type of a phenylalanine nanocylinder, which ultimately leads to nanofibril network. And apart from this SAC studies, we also did some computational analysis where we could get that actually this is hydrogen bonding and pi-pi stacking. So actually, to be very honest, I think one of the students who work upon this is Prabjot Singh. He might be here. So we were really excited about this work because this was a understanding of the phenylalanine network. If you go to the paper, you will understand it much better. So we are actually telling that how exactly these nanofibrils are getting formed by small monomers. There is a group in Israel which is working upon this. They have done a lot of studies on this. A group from this Professor Gezit is working on this. They have also identified different monomers in self-assembled structures. But of course, phenylalanine was done by us. So the nanospheres that you see, the small spheres, are actually the subunits. And these subunits are making the nanofibrils. And we have now started working on peptides also in this. Yeah.
Can you control how they assemble?
Yes, depending upon what type of condition you're using. So this nanofibril is based on a particular experimental condition, which is reported in the paper. And but also depends upon the nature of the monomer. If a monomer is able to interact effectively in different solvent systems, you can have multiple self-assembled architectures. Whereas if the monomer is not able to interact that much, depending upon what type of chemical constituents are there, then it is not possible for. Yeah. So yeah, I can send you a review article which talks about this.
Okay, thank you. I heard, by the way, I heard about Chandigarh is very clean now because of the coronavirus.
There is Chandigarh is having about 200 cases so far, and out of them it is localized only in one area, which has been about 150 of the cases are there. But relatively speaking, yes, Chandigarh is better than many other parts of India.
No, I don't.
I was talking about the air pollution.
Oh,
yes,
okay.
That's right.
Yes,
it is already. I mean, generally it is clean, but now it is like crystal clean. I mean, yeah, that's.
Really?
But we are going to pollute it very soon. Don't worry.
Okay.
Yeah.
It's much better than when you came. You came in November. At that time, it was quite polluted.
Yeah, it was heavy.
Yeah,
it will be back to normal, hopefully. Yeah. Unfortunately. Thanks a lot for joining with us. And it was very, very nice that you asked your question. So now that we are running almost out of time. So I would like to thank Rohit for giving a fantastic talk and making us aware of the research he is doing at Punjab University. And it was very, very nice listening to you. And I hope the students who have joined, they could make a good idea about Rohit's research. And in the end, I would like to thank the Chemical Society students who are taking up this initiative, Chemical Society GNDU. And next week, we will be having Dr. Salunke from your department only with us. So he will be talking about his research that he has been doing at Punjab University. The idea of this initiative is to make the students aware about the research which is being done in Amritsar or in the cities which are surrounding cities, or maybe in all over India also. But our main focus is to get the students know about the research which is happening around them so that they can make an informed decision. So all our sessions are recorded and they are available on YouTube channel, Chemical Society GNDU channel, and you can watch it. And Rohit will be uploading your session also there, so you can share it with someone if you want it. And thanks a lot again. And once again, I just thank you for doing this thing.
I think I really appreciate the way you guys have started this and the way this has enabled us to reach to an audience which is not possible in this time. So
yeah,
amazing. And I was really impressed. I think the BSc students are taking care of the
yeah. So the guy who has started it is Yatin. He is BSc second year student. So, and the one girl who is moderating it right now is BSc first year. So it is actually, so if there are some technical glitches or any other glitches, so they, I really am proud of that. At least they are doing it, and it is really, really great. And it is giving them all the confidence to talk with people. So last Monday we had Dr. Arunan from IISc Bangalore. So they themselves connected with him and requested him to give a lecture, and he came and they only manage it. I only jump in when there is some network issue, because otherwise it becomes formal. So thanks a lot, and thanks a lot students for organizing this, and I will see you again most likely next week. All right.
Thank you very much. Thanks a lot for having me.